Dr. Priyanka's biology, where we discuss everything from biology to biotechnology. So we had started with this chapter, that is the molecular basis of inheritance, from 12th standard NCERT textbook of biology. So here we had focused on DNA, what is DNA, the nature and composition of DNA, what are the biomolecules who contribute to this DNA. Then majorly we had focused on the double helix model or the salient features of the double helix model of the DNA proposed by Watson and Crick. So while studying this, we were studying that DNA is the actual biomolecule that is responsible for inheritance. But it was not a direct study that led to the conclusion that it is the DNA that is responsible for inheritance. But rather, there were many in between steps where people tried to prove or come to the conclusion that it is DNA and not any other biomolecule that is responsible for this inheritance. So we all know that father of genetics, that is Gregor Mendel, who started this study, and after him there were many people like Walter Sutton, then Thomas Hunt Morgan, then Miescher. They were proposing different things at from time to time, but it actually took a lot of time from the time it started with the study of genetics to the conclusion that it is the DNA which is the actual biomolecule responsible for inheritance, or the it is the actual genetic material. So in between there were many people, they were working. So the efforts of Gregor Mendel, then Thomas Hunt Morgan, they brought us to a conclusion that it is something to do with the chromosomes, and these chromosomes are located in the nucleus of the cells. So till here we had come to this conclusion. We knew a lot about all these things, but still that it is the DNA that is the genetic material, this particular fact was not proved. So a scientist, a British scientist called as Frederick Griffith, he was working on Streptococcus pneumoniae, and he was actually working towards the development of a vaccine for this Streptococcus pneumoniae, which is actually, or which was the causative agent for pneumonia. So this particular scientist, that is Frederick Griffith, he was working on all these things. So he said that, okay, I have to start working towards this development of vaccine now. So what he did, he was working on two closely related strains of Streptococcus pneumoniae, that is the R strain and the S strain. Now this R strain stands for a rough strain, and S strain stands for a smooth strain. So what is the difference between these things, that is the S strain and the R strain? So they were both were very closely related. So what he did was, as he was working, okay, this R strain, why this name comes, it is because of the rough surface. It had a very rough surface, and the smooth one had a smooth surface. So why was this happening? Okay, why there is difference in the R strain and the rough strain and the smooth strain? So smooth strain had a polysaccharide capsule, whereas the R strain had some, okay, it had a rough surface because it lacked this polysaccharide surface. Now because of the smooth nature and presence of this polysaccharide around the bacterium, that is the Streptococcus pneumoniae smooth strain, it was, or it always escaped the host immune system. So because of which it always remained alive, and it caused the disease, that is pneumonia, and the animal or whoever got it probably would die. Whereas the R strain, okay, being rough, it did not have the polysaccharide coating over it, so it was being attacked by the host immune system, and the strain used to die. So our Griffith, he was working with he was working with these strains of bacteria, and also he was using rats for these studies, mice for these studies. So what he did was, as he was studying, he isolated the rough and the smooth colonies of bacteria, and he injected them in different mice. So he saw that the R strain, when it was injected into a mouse, the mouse did not die because it did not develop pneumonia. Okay, whereas the smooth strain, okay, what it did, when it was injected into a mouse, it was seen that it caused pneumonia and the mice or the mouse would die. So he saw all these things, and then what he did was he he tried to analyze his studies further. So what he did was he heat killed. Okay, that is, he tried to kill those bacteria or that S strain by heating it. So that is called as heat killed. So he heat killed the bacteria and injected into the mouse. So to his surprise, he saw that the mouse was still alive because the bacteria were dead and they were injected and it caused no pneumonia. So the animal was safe and it survived. Now further what he did was he took the heat kill bacteria and he mixed them or inoculated with the live rough bacteria. He prepared a mixture of it and injected into the mouse. Now to his surprise, he saw that the mouse was dead. But actually, if this was the situation, if this was the earlier situation, okay, then the mouse shouldn't have died because he'd killed the bacteria S strain. So according to him, all the bacteria, the S strain, should have died, and the mixture whatever we have, that is the heat killed bacteria and the live bacteria, only the live bacteria should be alive and it shouldn't have killed the mouse. But what had happened was it had killed the mouse. So why it could have happened? What was the reason behind this? So he started analyzing it. So what he did was he collected or he isolated the bacteria from the dead mice. And to his surprise, he saw that he found live S strain bacteria. So how is this possible? That was the puzzle. Okay, because he did not understand what had actually happened. Okay, so what he did, he started analyzing this, whatever he had obtained from the dead bacteria, dead mice, and then he said that, sorry, okay, so then he said that something must have happened or somehow, okay, somehow the R strain must have got changed. So why that happened? What is that somehow? So all these things, questions, they will still remain unanswered for him. So further what he said that there is something that was transformed by this heat kill bacteria. So something, some jadoo tona must have happened and that changed that. Okay, मेरी आत्मा अंदर उसकी आत्मा बाहर, something of that sort must have happened and that changed the nature of this R strain bacteria. So he said, or he called it as the transforming principle. Okay, because transform is to change. Okay, so he said, okay, there could be something because something has happened now, because this R strain, it was not harmful. Okay, it was a mild one, but in spite of killing or hit killing the S strain bacteria, still somehow he could obtain the live S strain bacteria from the dead mice. So he is saying, he said that probably there is something that has changed or something that has transformed the R strain. Okay, something has changed and he called it as the transforming principle. Okay. So later on what he did was he started studying all this and he said that this could be because of the transfer of the genetic material. But however, he did not prove the actual biochemical nature of genetic material. So for this, further some other groups started studying and they established the fact that it is the DNA that has caused or that has changed that R strain, and further that led to the establishment of the fact that it is the DNA which is the actual genetic material. So we will focus on the experiments that was conducted by the other group, that was by Avery, McLeod and McCarthy. So that we will study some in some other videos. So till then, keep studying and keep smiling. And if you would like to take some of our assistance in your biology studies, please contact us. We will be always be happy to help you. Thank you.